Our bodies consist of trillions of cells. These cells need to divide when our bodies grow, replenish the cells we've lost from injuries, or need to maintain cell populations like that of the skin. The mitotic cell cycle is the process by which all eukaryotic cells replicate and divide to produce daughter cells for growth, repair, and development. Because the cell cycle is vital to our survival, our cells tightly regulate the process to prevent uncontrolled cell division or incorrect DNA replication, which can lead to diseases like cancer. This video gives an overview of the steps within the cell cycle and introduces some of the key proteins that help our bodies regulate cell division. We will use language that is defined in our cell cycle terms video, so we recommend that you watch that video if you haven't already done so. How exactly does the cell cycle enable DNA replication and cell division to produce more cells? We divide the cell cycle into 4 main phases: G1, S, G2, and M. We group these phases into 2 more general phases: interphase, during which the cell prepares for cell division, and the mitotic phase, when cell division occurs. In experimental assays, populations of cells can be either synchronous or asynchronous. A synchronous population of cells contains cells that are all in the same phase of the cell cycle. For example, a cell culture with many cells that are all beginning mitosis simultaneously. Cells in an asynchronous population are spread throughout the cell cycle stages. For example, some cells will be in S phase, while others are in M phase and actively dividing. This video focuses on one phase of the human cell cycle at a time, paying specific attention to how much genetic material is in the cell at each point. Note that when cells are not dividing, they often enter a quiescent stage called the G0 phase. These cells are performing their unique functions, but are not undergoing cell growth or division. Because these cells are not dividing, they will have 46 homologous chromosomes, or 2N chromosomes, no sister chromatids, and 2C DNA content because the cell is diploid and no DNA replication has occurred. The G1 or gap1 phase involves cell growth and preparation for DNA replication, which will take place in S phase. This preparation includes checking for cells with DNA damage, making sure there are enough nutrients in the cell for cell division, and verifying that the cell is big enough to divide. During the G1 phase, the cell also prepares for initiation of DNA replication. Specifically, proteins identify origins of replication and load multi-protein complexes onto this DNA. For example, the DNA helicase. How many chromosomes are currently in the cell after G1 phase? Try to fill out this table. Right now, the diploid eukaryotic cell has 2N chromosomes, 46 in humans. The cell modified the DNA to prepare for DNA replication, but no replication has occurred, and therefore the number of chromosomes and genetic material is still the same as in G0. That is, sister chromatids have not formed, and the cell has 2C DNA content because the cell is diploid and no DNA replication has occurred. At the end of G1, there is a point of no return, often called the restriction point, where the cell commits to completing the rest of the cell cycle and transitions into the S phase. DNA replication occurs in the S, or synthesis phase. The cell now activates the DNA helicases that the cell loaded onto DNA in G1 phase. This process separates the double-stranded DNA into single-stranded DNA to provide the templates for DNA replication. The sites of helicase action are junctions between double-stranded and single-stranded DNA, which form replication forks. Many other proteins assemble at these sites, like DNA polymerase, primase, and others, to replicate the DNA strands. The cell does not load helicases during S phase to ensure that the DNA is replicated once and only once. DNA replication results in duplicated chromosomes, sister chromatids that are held together by proteins that promote sister chromatid cohesion. Remember that sister chromatids are identical copies of a single chromosome. Now, how many chromosomes are currently in the cell after S phase, and how many copies of the DNA? Try to fill out this table. Right now, the cell is still 2N with 46 unique homologous chromosomes. The DNA replication duplicates each of these individual chromosomes to form 46 pairs of identical sister chromatids. And there is 4C DNA content now, because DNA replication has occurred in this diploid cell. G2 phase is a gap phase that allows the cell to prepare for M phase, during which the cell will divide in two. This preparation mainly includes verifying that S phase completed correctly and that the chromosomes were completely and accurately replicated, with no need for DNA repair or further DNA replication. Now, how many chromosomes are currently in the cell after G2, and how many copies of the DNA? Try to fill out this table. Right now, the cell is still in the exact same state as it was at the end of S phase. There are 2N, or 46, unique homologous chromosomes that have been duplicated to form 46 pairs of sister chromatids. Because DNA replication occurred in S phase, there is still 4C DNA content in the cell. Chromosome segregation occurs during M phase, or mitosis. We divide mitosis into subphases: prophase, metaphase, anaphase, and telophase, directly followed by cytokinesis, or cell division. Prophase is when the pairs of sister chromatids condense to form the familiar chromosome shapes we see in textbook illustrations. The nuclear envelope in the cell also breaks down. To simplify these animations, we only show 3 of the 46 pairs of sister chromatids in a human cell, but keep in mind that the remaining pairs are still there. Metaphase is when the mitotic spindle forms. This structure is a molecular machine that separates the sister chromatids, ensuring that each daughter cell gets one copy of each chromosome. During metaphase, the filaments, microtubules, from the poles of the mitotic spindle, centrosomes, attach to the pairs of sister chromatids on both sides at the region called the centromere. The tension from mitotic filaments pulling the pairs of sister chromatids toward the opposite poles of the mitotic spindle aligns the chromosomes in the center of the cell. This is called the metaphase plate. Anaphase is when the sister chromatids separate and are pulled to opposite sides of the cell by the mitotic spindle. Now, each side of the cell, which will become one of the 2 daughter cells, has a complete set of 46 chromosomes. During telophase, the nuclear envelope begins to reform around each group of segregated chromosomes. These chromosomes also begin to decondense once more. Cytokinesis is not technically a phase of mitosis, but follows immediately afterward. During cytokinesis, the plasma membrane of the mother cell fuses in a way that physically separates the cytoplasm of the mother cell to form 2 daughter cells, each with their own nucleus and chromosomes. Now, how many chromosomes are currently in each daughter cell, and how many copies of the DNA? Try to fill out this table. Each daughter cell is 2N with 46 unique homologous chromosomes. The pairs of sister chromatids have separated and are now just considered to be individual chromosomes in each of the daughter cells. Each new cell now only has 2C DNA content, like all diploid cells. Now the cell cycle is ready to start all over again with each of these daughter cells, or the cells can stop growing and arrest in the G0 phase. The cell cycle involves many steps, all of which are equally important to ensure that the replication of genetic material and cell division goes correctly. Now that you're familiar with the phases of the cell cycle, let's talk about what regulates these phases. How does the cell know what phase it's in, and how does it know that it's safe to proceed to the next phase? The cell cycle is regulated by a class of proteins called cyclins, which in turn activate cyclin-dependent kinases, or CDKs. The name cyclin reflects their property of fluctuating in abundance in specific ways during the cell cycle, as you can see in this diagram. The abundance of cyclin and cyclin CDK activity divides a cell cycle into the distinct phases we described before: G1, S, G2, and M. In addition, there are checkpoints during the cell cycle that check the work of the cell and ensure that one phase is complete before the next is started. For example, there is a checkpoint that prevents cells from entering mitosis until DNA replication is complete. This prevents catastrophic errors such as segregating incompletely replicated chromosomes, which would lead to chromosome breaks and DNA damage, which could in turn lead to diseases like cancer. The cell cycle is essential for our growth and survival. The cell cycle can appear complicated because it integrates many smaller, equally complicated cellular processes, such as DNA replication and mitosis. Now that you've finished this video, can you explain the 4 different stages of the cell cycle and what occurs in each step? And do you have an idea of what role cyclin and CDKs play in regulating the cell cycle and why they are important to our overall health? Thanks for watching!